There is a door in Hebron that no scientist has been allowed to open for 700 years. What happens when modern DNA technology gets close enough to listen? This stone monument, known as the Cave of Machpelah, represents one of the most highly contested sacred spaces in the world, revered by Jews, Christians, and Muslims alike. At its base lies a complex network of subterranean chambers. Around 19 BCE, Herod the Great enclosed these underground tombs within a massive superstructure of giant ashlar blocks. For centuries, pilgrims and conquerors walked above and below these stones. However, during the Mamluk conquests of the 14th century, the entry points to the inner caves were permanently sealed. Since then, the subterranean chambers have been entirely off-limits to direct scientific observation. This strict prohibition has effectively transformed the site into an isolated capsule. More than 3,000 years of continuous human activity remain compressed here into a single, undisturbed archaeological column. Because direct excavation inside the caves is forbidden, researchers must treat the site as a sealed forensic puzzle. A potential opening reportedly occurred in 2023. Reports surfaced of brief, limited access to sediment and bone material from areas immediately adjacent to the sealed chambers. If verified, this sampling event would offer a generational opportunity to extract physical biological evidence from the periphery of a site that has been closed to scientific inquiry for 7 centuries. To extract genetic information from a site like Hebron without disturbing its graves, researchers rely on sedimentary ancient DNA. This technique retrieves biological signatures directly from the earth, bypassing the need for skeletal remains. Organisms constantly shed microscopic cellular material into their surroundings, where it can bind to clay and mineral matrices. In 2021, a study published in Science by geneticist Eska Willerslev showed that these soil-bound strands can survive for up to 400,000 years. Willerslev's work established a rigorous methodological benchmark, proving that even highly degraded nuclear and mitochondrial DNA could be recovered from cave deposits through precise chemical extraction and strict contamination controls. For the reported Hebron study, researchers extracted sediment cores from stratified layers corresponding to the Middle Bronze Age, a period spanning roughly 2000 BCE to 1550 BCE. Analyzing these compressed soil samples requires specialized cleanroom environments and biochemical protocols designed to filter out modern contaminants. In these specific soil cores, researchers detected fragments of human mitochondrial DNA. The presence of these short, damaged sequences indicates that readable human biological residue survived within the compacted sediment of the periphery. Because these mineral structures protect the microscopic fragments from environmental decay, they preserve a localized genetic record of who occupied the space during the Middle Bronze Age, long before any modern excavations began. This biological footprint offers a direct window into the ancient populations of the southern Levant, bypassing the physical barriers that block direct access to the tombs. Rather than pointing to a specific historical individual, the mitochondrial DNA recovered from these soil layers corresponds to a broader population history model. This genetic signature matches the Proto-Semitic ancestral cluster defined in a landmark 2020 study by geneticist David Reich. Published in the journal Cell, Reich's framework maps the genetic makeup of the southern Levant during a specific chronological window between 2200 BCE and 1800 BCE. This ancestral profile is characterized by a distinct genetic mixture, combining ancestry from Anatolian Neolithic farmers with populations from the Iranian Chalcolithic. Archaeologically, this combined signature is closely associated with ancient Canaanite populations of the Middle Bronze Age. Genetics shows that this ancient baseline did not vanish. The same broad ancestral signature appears today in the founding lineages of modern Middle Eastern populations, including Ashkenazi Jews, Arab Palestinians, and certain Coptic Christian communities. Supporting this biological thread, a 2022 study from the Weizmann Institute of Science determined that modern Jewish populations inherit between 40 and 65% of their ancestry from these Bronze Age Levantine groups. Yet geneticists draw a strict distinction when translating these findings. Ancestry resemblance remains entirely separate from proving the identity of any single named biblical figure. These biological signatures document the genetic background of the collective populations who moved through, settled, and occupied the southern Levant 4,000 years ago, rather than identifying a specific historical patriarch. Beyond the microscopic traces suspended in the soil, the reported 2023 access also yielded a physical specimen, a single bone fragment recovered from a subterranean chamber adjacent to the main tomb. This fragment underwent accelerator mass spectrometry radiocarbon dating, a precise method that measures carbon-14 decay within the preserved bone collagen. The analysis returned a chronological range from 1880 to 1650 BCE, established at a 95% confidence interval. This precise timeframe corresponds to the Middle Bronze Age, the exact historical era traditionally associated with the patriarchal migrations described in ancient Near Eastern texts. Geneticists succeeded in extracting ancient mitochondrial DNA from the dense bone matrix, assigning the specimen to haplogroup J1b1a. This maternal lineage represents a highly specific genetic branch passed down exclusively from mother to child, mapping a maternal line of descent. In historical and modern databases, this subclade clusters primarily within populations historically associated with ancient Mesopotamia and the southern Levant, particularly Canaan. Dr. Gila Kahila Bargal, a prominent ancient DNA expert from the Hebrew University of Jerusalem, acts as the lead researcher evaluating these findings. Bargal has described the convergence of these three distinct elements: the specific geographical find spot, the Middle Bronze Age radiocarbon date, and the Mesopotamian-linked maternal haplogroup, as statistically improbable to occur by mere chance alone. However, the research team maintains strict scientific boundaries. A highly unusual genetic convergence does not constitute proof of a specific biblical individual. This single bone fragment cannot be definitively linked to a named historical patriarch. Instead, it serves as a rare, tangible biological anchor, proving that the genomic signatures of Middle Bronze Age populations remain physically preserved at the periphery of this highly restricted site, offering a direct and objective look at the regional gene pool. The genomic profile of the Hebron-adjacent specimen aligns with a massive genetic corridor stretching across the ancient Near East. A 2019 study published in Science Advances analyzed genomes from the ancient Mesopotamian city of Ur, revealing a population with significant Iranian Chalcolithic ancestry. Further north, genetic analyses from Haran identified a transitional population carrying a distinct mixture of Anatolian Neolithic and Mesopotamian genomic signatures. This exact genetic pattern recurs at major archaeological waypoints along the geographic routes described in early Semitic historical texts. Between 2021 and 2024, 4 peer-reviewed papers in Nature Genetics and the American Journal of Human Genetics mapped these Bronze Age population shifts. These studies demonstrate that the physical movement of populations from Mesopotamia into the southern Levant occurred as a large-scale regional migration, permanently altering the genetic landscape of the ancient world. This genetic thread connects directly to modern populations. A 2000 study by Michael Hammer in the Proceedings of the National Academy of Sciences identified high frequencies of haplogroup J across Jewish communities, confirming a shared Middle Eastern ancestry. In 2021, research in Current Biology indicated that Jewish and Arab ancestral populations were genetically indistinguishable 3,000 years ago. This supports Johannes Krauss's framing of how ancient texts can preserve population memories later tested by genetics. Yet, scientists maintain a strict interpretive ceiling. Although Hebron soil may preserve DNA for 4,000 years, a single bone fragment cannot validate theological narratives. Mitochondrial sequences only trace maternal descent, not complete autosomal genomes. High contamination risks and small sample sizes mean genetics can document regional migrations, but never prove the existence of individual biblical figures. Today, science stands at the threshold of the sacred, reading history written in the soil. As genetic technology advances, these microscopic sequences challenge modern divisions, reminding us that the deepest roots of this region are inextricably shared. What do you think? Share your thoughts in the comments.